Good morning. My name is Aleyda Acosta. I am a postdoctoral research associate at the University of Florida Gulf Coast Research and Extension Center. Thank you for your interest on my talk. The title of my presentation is Improving Crop Management for Hop Production in Florida, Effects of Plant Density and Nitrogen Rate on Plant Growth, Yield, and Corn Quality. Hops are important to provide stability, bitterness, aroma, and flavor to beer. The cons of the hops is the reproductive structure of this species that has thousands of lupulin glands that give the characteristic yellow color to the beer. These lupulin glands contain alpha acids, beta acids, and essential oils, and the profile of these chemicals vary among the cultivars, which are used to produce different types of beer. The hops and the craft beer industry has increased in the United States for the last 20 years. The plot in the left shows the hop production in United States. The blue bars are showing the production from 1998 to 2018 in millions pounds. The difference here is that 20 years ago, United States were producing 60 million pounds, and by 2017 is producing more than 100 million pounds. The grower price has also increased and is represented by the orange line. From 1998 to 2006, the price for each pound of dry hop was less than $2. By 2017, is now near $6 per pound. The craft beer industry represents 12% of the United States beer industry by volume. And in Florida, this expanse of the craft beer industry has been significant. For example, the number of breweries passed from 45 in 2011 to 329 in 2019. The craft beer production also increased from 5.4 million gallons in 2013 to 43 million gallons in 2019. And the economic impact, of course, increased from $876 million in 2013 to $3.6 billion in 2018. The map shows where the hops grow. So this plant is from the temperate areas. The green areas are the areas where a commercial production is made. The yellow range shows the median that between 33 and 55 degrees latitude north and also south is the ideal location to produce hops. Florida is outside that range. Florida is in subtropical areas, and we have made some important modification to make that possible, to make hops grown in Florida. Hops has a specific requirements. They are short day plants, and they require at least 15 hours of day length to keep the vegetative growth. Flowering is induced when day length is lower than 15 hours. Hops also require a trellis to sustain growth and yield. And in Florida, we added a supplemental light system to meet the hops photoperiod requirements. This is a picture that shows how plants look at night under supplemental light system. So we are artificially increasing the day length of hops by more than 15 hours to keep the vegetative growth in Florida conditions. By doing that, we are able to sustain 2 growing seasons per year that we call spring season and fall season. This slide shows the process for one of the season, the season in spring. When the plants are at least 3 feet tall, we turn on the lights, and that keeps the plants growing in a vegetative stage. So the plant have the chance to grow, to rise the vines. The vines also develop laterals, and this process happen over 42 days. After that, we turn off the light, and just 7 days after that, the vegetative buds turn into inflorescent buds. 13 days after, they have flowers, and 19 days after turn off the lights, we have cones in develop. The process from turning off the lights until the time that we are harvesting is more or less 2 months. So we can control when we want to harvest. So by doing that, we harvest in June, by the end of June, and we also harvest by the end of December. Uh, After we succeed with this system, the next step was to try to improve yield by making experiment with crop management practices. So the goal of the research that I will show you was to identify the effects of hill spacing and nitrogen application rate on growth, yield, and cone quality of Cascade hops growing in Florida over the first 2 years, 2018 and 2019, and of course the two growing seasons from each year. Inside the methodology, we were using a split plot design with 3 hill spacing and 4 nitrogen rates. The hill spacing were 76, 91, and 107 centimeters. The nitrogen rate were different rates between the seasons, but they conserve, or what is common of them is the percentage of change between the treatments were similar. For example, in all seasons, nitrogen 1 was 27% of the maximum concentration. Nitrogen 2 was 52% of the maximum concentration. Nitrogen 3 was 76% of the maximum concentration, and nitrogen 4 was, of course, the maximum concentration. We study this effect in hill spacing and nitrogen rate by 2 years, year 1, 2018, and year 2, 2019, 2 seasons per year, spring and fall, for a total of 4 seasons. We used different ways to measure the effect of this nitrogen and hill spacing. The picture in the left side show 2 hills. For those who are not familiar with hops, we have there 2 hills. Each hill has 2 twines that support the growth of several vines inside each twine. So we measure the length of the vines. We measure the number of vines. We also measure the number of branches, and we harvest the cones. We dry the cones to 10% moisture. This is a standard protocol for hops, and we measure that dry hop. We also send some sample for analysis to analyze alpha acids, beta acids, and total oils. In the results, this is the growth of the vines, the vine length among the years for spring in purple color and fall in orange color for year 1 and year 2. In general, we didn't find a difference. What we can see is that there is a fast growing season, and that is common for all of them, and that is it shows that hops can grow, the vine can grow as fast as 13 centimeters per day. This is the growth of the vines, the number of vines. We are measuring the effect of nitrogen nitrogen application rates over the number of vines. We didn't find difference in spring year 1 or fall year 2. We did find difference in year 2 spring. The difference is that nitrogen 1 is different from the other nitrogen rates application. We didn't have data for year 1 fall. For the number of lateral shoots, the branches, we find difference in the year 2. So nitrogen The lower concentration of nitrogen produces less number of branches in year 2. We did not find any difference in year 1 spring. We have no data for fall in year 1. Something that caught our attention is that nitrogen 1, as you see in the picture, has also the branch has less leaves and less cones in general. Now we pass to production. The plots are showing the dry cone yields in kilograms per hectare and how they are different between year 1, year 2 in blue colors, and also for spring and for fall. In year 1 and year 2, we can see that the year 2 has more cones, 49% more cones in general, and comparing spring and fall, we saw a decreasing of production. So fall has between 26 and 37% less cones. When we analyze the effect of the 4 nitrogen rates on the yield among the 4 different seasons, we found that in general, the more nitrogen we apply to the system, the more yield we get. For the year 1, spring and fall, we find a linear trend of that increase, a positive increase, and for year 2, we found more difference. The ANOVA showed significantly difference among the treatments in year 2, spring and fall. We also have a significant trend, linear trend for fall, and a quadratic trend for year 2. But in general, all of them, the more nitrogen we apply, the more yield we get. This is a picture of how the hops look 1 day before harvesting. This is the cone quality. So we analyze alpha acids, beta acids, and total oils and difference between year 1 and year 2, and also differences between spring and fall. So in general, alpha acids and total oils are lower in year 2 comparing to year 1. And alpha acids and beta acids are lower in fall comparing to spring. This is the effect of nitrogen application rate on alpha acids. In the y-axis is the alpha acids in percentage, and in the x-axis you see the 4 nitrogen rates for each season, year 1 spring fall and year 2 spring fall. In general, we saw contrasting results. For example, year 1 in fall, there is an increment of alpha acids as we increase the nitrogen, but we got that totally opposite behavior in year 2 spring. For beta acids, this is the same type of plot that the one before. We just found that in year 2 there is a decreasing of the beta acids content as we increase nitrogen application rates. And for total oils, we see that in year 2 spring also decrease, the total oils also decrease in year 2 springs, and we increase the nitrogen application rates. So as a conclusion, we can say that Florida can produce two growing seasons per year. So we have 2 harvests. The growing season, we call it spring, that go from February to June, and July, that go from July to December. Hops production differ between years and seasons. We found an increase of 49% in year 2 compared to year 1, and also yield can decrease 26 to 37% from spring to fall. Yields show in general a positive linear or quadratic trends when increasing nitrogen application rates, and we found that hop quality is greatly affected by seasons, having at the moment inconclusive results regarding nitrogen application rates. With that, I would like to thank the University of Florida, the Golf Course Research and Extension Center, the Horticultural Lab heading by Shinsky Agihara, for the sponsors and collaborators that contribute to this project. With that, I would like to thank and I'm open to any questions. Thank you.